And here we are, myself, Brian, Andreas Hellander, Fredrik Wrede, and Prashant Singh. Okay, so here's the schedule. I'm going to begin with just a brief introduction to stochastic simulation and a fairly wide range of problems that we've used this software on. And then Brian's going to take over, and he's going to teach you about using StochASS Live. In tomorrow's session, it'll be about mostly about our new capabilities for model exploration and parameter inference in StochASS Live. And Andreas and Fredrik and Prashant are going to present in the first part. And then in the second part, Brian's going to teach you about some advanced topics. Okay, so the StochASS, we call it the family of software. StochASS Live is a software as a service platform to support modeling. It enables you to create, simulate, infer, and explore both deterministic and or discrete stochastic models. And literally, you don't really have to do a thing to get started with it. And Brian will show you that. Now, if you want more hands-on, then our computational engines are also all available separately. So Gillespie 2 for stochastic simulation of well-mixed systems, Spatial Pi for stochastic simulation of spatially dependent systems up to 3D, and SIOP for parameter inference and model exploration, exploring the parameter space. Okay, so I want to just sort of set the stage for the kinds of problems, the kind of mathematics that StochASS does and does not address. So first, I want to draw the distinction between extrinsic stochasticity, which we're not addressing, and intrinsic stochasticity, which we are. Okay, so extrinsic stochasticity is just noise arising from random events in the environment. And usually this is modeled by some kind of Gaussian distributed or white noise. And it's often modeled via stochastic ODE or PDE system. This is not what we're doing, okay? We're doing intrinsic stochasticity, okay? And that is noise arising due to small populations of key players, okay? And this plays a big role, for example, in cell biology, and where the transcription factors that communicate with the genes are often present at any given time in extremely small numbers, and which leads to a lot of stochasticity, can actually change what your solution is doing, what it looks like. It's major. And it's also a big thing that happens in ecological models when, for example, species are going extinct. Okay, so, and this is modeled as a Markov or past forgetting process. Okay, and this sort of intrinsic stochasticity really took off when this famous paper by Michael Elowitz was published in Science, where he revealed by an elegant experiment that there was intrinsic stochasticity in cell biology, that it was playing a major role, I should say. Okay, so ecologists have known that for a long time. okay, that intrinsic stochasticity is really important to take into account. And of course, the big place where it comes into play is extinction. Okay, once you get down, so think about it, an ODE model or PDE model can't how should I say, nothing can go extinct. Okay, it could become smaller. The populations could be smaller and smaller until you have, say, here's a frog, until you have a millionth of a frog or something like that. But somehow frogs could re-arise from that. Okay, whereas in the stochastic model, when it goes down to zero, that's it. That's the end. So Brian and I and some other people did some interesting simulations with StochSS a while back, where we modeled communities of frogs, okay, which are going extinct due to the chytrid virus. They're going extinct around the world, all different kinds of frog populations. And we modeled possible interventions that ecologists were either taking or thinking about taking, or, this gets to be a desperate situation. Okay, another interesting sort of stochastic situation that I came across was in medicine. And one place in medicine that this occurs is in the coagulation cascade. Okay, so like when you prick your finger, okay, and your blood starts clotting. Okay, but how long is it going to take for the blood to start clotting? Of course, this differs across individuals, but it also differs quite a bit with just you. If you were pricked in the same place or whatever. Because it's a long chain of reactions that have to happen, and the early ones involve fairly small populations of key chemical species. Okay, so the early part of this cascade is really quite random. This is my favorite one. This is it has to do with circadian rhythm. And this is what we call the black line is what we call a phase response curve, okay? And that's been around for quite a while with ODE models. And if you just think of circadian time, so 24 hours, and that line of 4 as the time when you wake up, okay? And that line out there at around 22 as sort of the deep middle of the night. Okay, then the phase response curve tells you, if you make a perturbation, say at 4, okay, how much can it change the phase, okay? And if you're just on the curve here, if you're doing a deterministic system, the system is really quite stable at the time that you usually wake up, okay? So a perturbation is going to, by the way, what I mean by a perturbation is like daylight, some idiot opens your blinds, okay? Who knows, maybe your spouse or whatever. And then, but anyways, later in the phase response curve, you can get, a perturbation down here at around 16 will move your phase in a negative direction the next time that after 24 hours, okay, you'll be thinking it's a different time. And of course, we all worry about phase response and phase response curves. We probably don't think of it that way. I do, but most of us probably don't think of it that way. But when we, take an international flight, I'm dying to do that again. I don't know about you, but maybe not the flight, but the destination would be fun. Anyways, if you take an international flight, it could be really bad. California to Europe is a few days of, some kind of misery, personal misery of not being able to sleep in the middle of the night, that sort of thing, right? What this shows, so this is a stochastic model of the circadian biology, okay? And what it shows is that there's a tremendous amount of variation to what can happen, how cells respond if it gets the perturbation just literally in the dead of night, in the middle of night when you're in deep sleep, okay? And as you can see, the cells are going all over the place. They're setting their faces in crazy places. Well, you can actually use this to reduce your jet lag, okay? So if you take a flight that arrives in your destination, at the time, which would have been the middle of the night at your starting point, okay? So for me, middle of the night in California, then your circadian cells in your brain are just gonna they're gonna go all over the place. You know, they don't know what time it is. Okay, and that's great because it's much easier, much faster to sync to the new time at the new destination if you're all mixed up than to just move a whole bunch of cells who think they know what time it is. Okay. And Brian and I have been involved in this problem for a long time. It's also been a really fun problem. It has to do with intrinsic stochasticity in cell polarization. So these are some yeast cells, and what they've polarized. Okay, so you can see there's the orange part there is showing what we call the polarisome. Okay, which is really the center of the action of the cell chemistry that's forming the projection. Okay, and you see the cell is kind of moving a lot of molecules to make this projection. Okay, it's a very strongly nonlinear effect that comes about basically due to stochasticity. And in this case, it's due to a relatively small number of actin cables in the vicinity of the polarisome. So we've modeled this guy in up to 3D. Up to, what else would we have? 4D. Okay, so it's time for me to be finished. Okay, another place where I've had an effort for a while, it's pretty fun, has to do with crowding in the nucleus. Okay, so human cells cram almost 78.7 inches of DNA into a nucleus that's about 0.0002 inches wide. Okay, and then a transcription factor, which turns on and off the genes, needs to go in there and literally, this is a good picture. I searched for the best illustration I could find. It needs to go into that mess and find the appropriate activated. And you can imagine that there's a lot of stochasticity involved in how long that's going to take. So I'm done with the introduction, and I'm going to hand this over to Brian. That means I have to stop sharing.
All right. And just to confirm, everyone can see my screen? So, okay. Perfect. All right. Yeah, so there's a lot of great applications that we can work on. So now I really want to talk about StochASS. What have we built here? So this is what we call our integrated development environment for simulation and analysis of a discrete stochastic biochemical system, biochemical models. Really, what we want to do here is put the power of modeling in the people who can benefit most from it, which is everyone. We want to get modeling out there into the community, and we want to remove the technical limitations of that and bring in some of the best things we can do. So yeah, okay. So what is our vision? Integrated development environment that lets you quickly build and simulate models. And we say stochastic, but ODE models work perfectly fine. We started off with biochemical, really, but epidemiological and ecological models have work equally well with the very same tools that we're using. So we're sort of expanding our environment here. Not just build and simulate those models, you need to visualize those results. And you need to have a rapid development cycle where you can see what's going on and go back and forth. And then once you've built your initial model, you need to explore the parameter space. What different parameters give you different mechanics? All of this really is just to gain insight from modeling. We want to publish papers. We want to do the science, and that insight is what we're hoping to provide and speed up the iteration cycle and move us forward faster. StochSS is a single piece of software that builds upon a set of softwares that Linda already mentioned already. The first one I'll mention is Gillespie 2. So this is what we do for well-mixed simulation. So for ODE, aka deterministic simulation, and also SSA for stochastic simulation. We've got our spatial pi software. So this does a spatial stochastic simulation algorithm. And it also does a particle-based fluid dynamics. So those two work together. And we've also got we've got some beta features in there. And one of them is the deterministic spatial simulation of biochemical reactions as well. And then we've got SIOP, the machine learning assisted inference, optimization, and parameter exploration. So those tools are the actual engines, and you can use them independently. They're each Python libraries that you can download and use inside of your own software, or StochSS provides a nice interface to use all of them directly. So the idea is that this is a powerful platform for computational experiments. So what is a computational experiment? Well, you have a problem, you specify that. So in the model editor, you're going to set up whatever your problem statement is. I have these variables, I have these rates, I have these things that change my variables around. Our simulation tools then allow you to take that model editor or the domain you define upon and run it using ODE, spatial stochastic, or the well-mixed stochastic or simulation methodologies. You can also take this same model and integrate it with data. So we're going to do some parameter estimation or look at the parameter sensitivity of the various things to give us some more insight into this model as we develop it. And at the end, you need to have some sort of output visualization tools, so plot tools, 3D visualization tools. And you'll see we've deeply coupled the Jupyter notebooks that allow you to expand upon this data and then process it independently. So the StochSS software ecosystem, I'm going to hit all these things again. So the StochSS itself is a user interface, a model building and visualization software. It uses the Gillespie 2 ODE for well-mixed simulation, spatial pi for spatial simulation, and SIOP for the inference. All of this lives inside of a Docker container. So that container provides a platform with all the necessary libraries and everything for you to run this on whatever system you may want to run. So that's really the StochSS ecosystem all in one. Now, StochSS Live is our software as a service presentation of StochSS. So you can see we have that same Docker container here, StochSS Live, but instead of just being one container, you have one container per user. All of this is done using the Jupyter Hub software platform that allows us to have user authentication, resource allocation, and scalability. All that's running on some sort of compute infrastructure that we have, which is both server hardware and some cloud resources coupled together. And this whole thing together is StochSS Live. So we'll be using this here in just a few minutes. You can also, of course, just run StochSS on your own laptop. And I'll show you how to do that as well. You can also set up StochSS on your own server and use your own hardware to run these computations. So we're trying to make this as flexible as possible and make sure that you can get this out in the hands of everyone. And really, that's why we have StochSS Live, to make sure that anyone who has a web browser can do these computations. So StochASS Live is a web app. So you simply go to live.stochass.org and you'll see this web page here. And just looking ahead, yeah, okay. So